Carbohydrates include both simple sugars, which are little ring shaped molecules made of carbon, hydrogen, and oxygen, either alone or in pairs, as well as more complex carbohydrates, which are formed when these rings link up together to make long chains. Carbohydrates provide us with calories, or energy, and simple sugars in particular play a lot of roles in our diet. They sweeten lemonade, balance out an acidic miso soup, fuel yeast in rising dough and alcohol, and help preserve jams and jellies. Now, sugars are found naturally in plants like fruits, vegetables, and grains, as well as animal products like milk and cheese. Added sugars are the sugars that get added to foods, like cereals, ketchup, energy bars, and even salad dressings. To be clear, even if the sugar being added comes from a natural source like sugar cane or honey, it's still considered an added sugar. In fact, a variety of ingredients listed on food labels might be sources of added sugars, some of which you're probably familiar with. Sugar actually refers to a family of molecules called saccharides. Monosaccharides, where mono means one, so one sugar molecule. Disaccharides, where di means two, so two sugar molecules linked together. Oligosaccharides, where oligo means a few, so it's 3 to 9 sugar molecules linked together. And polysaccharides, where poly means many, so it's 10 or more sugar molecules linked together. Glucose is the most important member of the sugar family, and it's a monosaccharide. It's one of the main sources of calories for the body, and is able to cross the blood-brain barrier and nourish the brain. Another monosaccharide is fructose, which is commonly found in honey, fruits, and root vegetables. Finally, there's the monosaccharide galactose, known as milk sugar. It's known as milk sugar because it's only found in nature when it links with glucose to form lactose, a disaccharide found in the milk of mammals, which includes cow milk as well as human breast milk. Sucrose, or table sugar, is another disaccharide, and it's formed when fructose links up with glucose. Sucrose is found in various fruits and vegetables, with sugar cane and sugar beets having the highest quantities. Maltose is another disaccharide, and this one is two glucose molecules linked together, and it's found in molasses, which can be used as a substrate to ferment beer. Sugars, like fructose for example, are most always found in combination with other sugars, and the combinations can be pretty different, even in seemingly similar foods. For example, in honey, 50% of the sugar is fructose, 44% is glucose, 4% is galactose, and 2% is maltose. Whereas in maple syrup, less than 1% of the sugar is fructose, 3% is glucose, and 96% is sucrose. So simple sugars, whether they're natural or added, are mixtures of monosaccharides or disaccharides. Next, there are the complex carbohydrates. There are oligosaccharides, like galacto-oligosaccharides, which are short chains of galactose molecules, like those found in soybeans. Then there are polysaccharides, which are even larger chains with branches, and are the most abundant type of carbohydrates found in food. Starches are polysaccharides with molecular bonds between sugar molecules that human intestinal enzymes can break down. Starches are an important source of calories, and can be found in foods like rice, potatoes, wheat, and maize. Starches don't taste sweet like simple sugars, because they don't activate the taste buds in the same way. And there are also dietary fibers, which are carbohydrates that intestinal enzymes can't break down, and so the body can't digest them. Now, there are many different types of dietary fibers, and they're not all the same when it comes to their structure or impact on health. Fibers have molecular bonds that are resistant to human intestinal enzymes, so they pass through the small intestine undigested, get broken down a bit by bacteria in the large intestine, and ultimately end up as bulk matter in the stool. Fibers are critical because they can slow down the rate of absorption of simple sugars like glucose in the small intestine, which can help maintain healthy blood glucose levels. They also increase stool weight, which helps prevent constipation. And fibers like beta-glucan are also good for heart health. Monosaccharides link together through glycosidic bonding, which is when an OH group from the carbon on one monosaccharide binds with the hydrogen from the carbon of another monosaccharide. Together, this forms an H2O, or a water molecule, which goes away. In the case of maltose, that leaves an alpha-1,4 glycosidic bond, which is a bond between carbon number 1 of one glucose monosaccharide and carbon number 4 of the other glucose monosaccharide. And alpha refers to the fact that the molecules are lined up next to each other. Lactose, on the other hand, has a beta-1,4 glycosidic bond, meaning that carbon 1 of galactose and carbon 4 of glucose are bonded, but this time the molecules are stacked with one higher than the other. Finally, sucrose has an alpha-1,2 glycosidic bond, meaning that carbon 1 of glucose and carbon 2 of fructose are bonded. Now, when you eat something, like a piece of onion bread, enzymes start breaking down the disaccharides, oligosaccharides, and polysaccharides into monosaccharides so they can be absorbed. Different enzymes help to break down different linkages. For example, amylases break down large polysaccharides like starch into smaller units, whereas lactase, sucrase, and maltase break down lactose, sucrose, and maltose into their monosaccharides. The individual monosaccharides that result from the digestion of larger carbohydrate molecules, glucose, fructose, and galactose, cross the gut lining and get into the bloodstream to get used by the body. When glucose levels in the blood increase after eating, the pancreas releases the hormone insulin, and this helps move glucose into all cells for energy. Insulin helps stimulate the liver to store glucose as glycogen in a process called glycogenesis, which is when some of the glucose molecules get linked together with alpha-1,4 and alpha-1,6 glycosidic bonds to form a polysaccharide called glycogen. Insulin also promotes fat and protein synthesis. Now, metabolism of galactose has an initial step where an enzyme in the liver converts galactose into glucose. It basically flips the orientation of the OH group on the fourth carbon. Just like that, galactose can become just another glucose molecule in the liver. Fructose, though, is handled a bit differently by the liver. Fructose has a total of 6 carbons, and most of it's broken down into 2 3-carbon molecules and sent into glycolysis to help generate energy. When energy is needed, all 3 monosaccharides are metabolized through glycolysis, then the citric acid cycle, and oxidative phosphorylation. Ultimately, all digestible carbohydrates, regardless of whether they come from simple sugars in honey or from starches in baked potatoes, are broken down into their component monosaccharides for immediate energy use or stored away for a rainy day, depending on what the body needs. The National Academies of Sciences, Engineering, and Medicine recommended that a healthy diet contain 45 to 65% of its calories from carbohydrates. The number of calories you need to maintain your weight depends on things like your age, sex, height, weight, and activity level. For example, let's take this slightly active 40-year-old woman who's 5 foot 9 inches and 160 pounds, with a BMI of 23.6 and requires a 2,000 calorie diet. And let's say she wants to aim for 55% of her calories from carbohydrates. So that's 55% of 2,000 or 1,100 calories. And there are different types of carbohydrates. First, there's fiber. The general recommendation is to get 28 grams of fiber in a 2,000 calorie diet. With roughly 0 to 2 calories per gram of fiber, that's about 56 calories, or about 3% of her total calories. Next, there's sugars, both the kind that are added to foods and those naturally found in whole foods. There are few formal recommendations for total or natural sugar intake. However, updated Canadian nutrition labels are based on a daily value of 100 grams or 400 calories from total sugars, which is 20% of a 2,000 calorie diet. When it comes to added sugars, both the World Health Organization and the US Dietary Guidelines recommend that they make up fewer than 10% of total calories. Just like her goal for total carbohydrates, she's aiming for the mid-range of the added sugars recommendation, which would be 100 calories or 5% of her total calories. Using this approach, the remainder of her total sugar calories, 300 calories or 75 grams, would come from the sugars found in fruits, vegetables, dairy, and grains. This would be 15% of our total calories. And that leaves 640 calories or 160 grams from starches, or 32% of our total calories, to reach our carbohydrate intake goal. Now, eating a healthy diet means choosing foods that are as nutrient rich as possible. And foods that contain fiber, starch, and natural sugars like fruits and vegetables tend to be richer in nutrients than those with added sugars. Having said that, processed and packaged foods are a part of most people's diets. So, carefully reading nutrition labels can help you compare foods and choose more nutrient rich options. Generally speaking, picking foods and beverages that are higher in nutrients like fiber and lower in added sugars is best. Alright, as a quick recap, there are various types of carbohydrates. Simple sugars are monosaccharides and disaccharides that the body can readily absorb. Starches are polysaccharides that take longer to absorb. And fibers are polysaccharides that the body can only partially absorb with the help of gut bacteria. Ultimately, a healthy diet includes all types of carbohydrates, coming from a variety of sources like fruits, vegetables, dairy, and grains. It can include added sugars too, with the World Health Organization and the US Dietary Guidelines recommending that added sugars make up less than 10% of your overall calories. Fats are an essential part of a healthy diet. They contribute to the taste and texture of foods, like the smoothness of guacamole and the flakiness of a croissant. Fats are also a major source of energy and a critical component of cells and tissues, and they also help absorb essential vitamins, and can be converted into other molecules like prostaglandins, which help cells communicate with each other. Fats have a 3-carbon backbone called glycerol, as well as fatty acid chains. The fatty acid chain is basically a string of carbon and hydrogen atoms. When an OH group from the glycerol molecule binds to a hydrogen from the fatty acid, an H2O or water molecule gets released, and the two molecules link up. If this happens once, the result is a monoglyceride. If it happens twice, it's a diglyceride, and 3 times makes a triglyceride. Now, there are various types of fatty acid chains, and one way to categorize them is by their length. In other words, how many carbons they have. Short-chain fatty acids have 2 to 5 carbons, medium-chain fatty acids have 6 to 12 carbons, and long-chain fatty acids have 13 or more carbons. Fatty acid chains are also categorized by the bonds connecting the carbons in the chain. A single bond is just one bond between the carbon atoms. And when a fatty acid chain has only single bonds, it's called a saturated fatty acid, because it has as many hydrogen atoms as possible, or it's saturated with them. Triglycerides with saturated fatty acids are nice and straight, so they pack together really well, and as a result they're usually solid at room temperature. And the longer the saturated fatty acid chain, the more likely it'll be solid at room temperature. Carbons can also have double bonds between them though, and when a fatty acid has one or more double bonds, it's called an unsaturated fatty acid, because it's not saturated with hydrogen atoms. For every double bond, there are 2 fewer hydrogen atoms. Also, a double bond causes a kink in the molecule, so the triglycerides don't pack together as nicely as saturated fats. As a result, unsaturated fats are usually liquid at room temperature. Unsaturated fatty acids can be further classified according to the number of their double bonds. Monounsaturated fatty acids are unsaturated fatty acids with just one double bond. Polyunsaturated fatty acids have 2 or more double bonds. Also, they can be classified according to their location as well. Since all these hydrogens can get kind of crazy looking, we'll just take them away for now. So another name for the methyl end is the omega end. And then we can count the number of carbons until the first double bond. Since this one is 3, it would be an omega-3 fatty acid. If the double bond is 6 carbons from the end, it's an omega-6, and if it's 9 carbons from the end, it's called omega-9. Now, to make things even easier when looking at these molecules, I'm just going to show the bonds. Alright, so omega-3s are usually polyunsaturated fatty acids, and include alpha-linolenic acid, or ALA, eicosapentaenoic acid, or EPA, and docosahexaenoic acid, or DHA. EPA and DHA are marine sources of omega 3s. They're produced by microalgae and end up in the tissues of fish like anchovies, mackerel, salmon, and sardines. ALA is found in plants like flaxseed, walnuts, and canola and soybean oils. Our bodies can convert ALA into EPA and DHA, but it's an inefficient process that yields only small quantities. And that's why dietary recommendations include foods that have EPA and DHA. Omega-6 fatty acids are also usually polyunsaturated and include linoleic acid and arachidonic acid. Linoleic acid is found in oils like safflower, corn, and soybean oils. Arachidonic acid is found in animal sources like fish, meat, and eggs. Our bodies can convert linoleic acid into arachidonic acid, but once again the process is inefficient. Because ALA and linoleic acid can only be obtained in the diet, they're considered essential fatty acids. Omega-9 fatty acids are typically monounsaturated fatty acids, and an example would be oleic acid, and these can be made by the human body. Foods like canola and olive oil, as well as almonds, contain omega-9s. Now, looking at the double bond of this unsaturated fatty acid, like most unsaturated fats, it's got a cis configuration. In a cis configuration, the two functional groups are on the same side of the double bonded carbons. Now, when this happens, the fatty acid chain naturally bends. A molecule that bends does not pack tightly together, so it's a lot more fluid. Think about cooking oils, which are liquid at room temperature. Some fats are in a trans configuration, though. In a trans configuration, the functional groups are on opposite sides of the double bonded carbons, and this keeps the chain more straight and easier to pack. Trans fats result from a process called partial hydrogenation. In just plain old hydrogenation, hydrogens are added to cis fats to get rid of all of the double bonds. So let's say that you've got this triglyceride, and it has a total of 2 double bonds. So in this case, we'd add 4 hydrogens, 2 for each double bond. That turns the unsaturated fatty acids with cis double bonds into saturated fatty acids. Partial hydrogenation, on the other hand, refers to adding hydrogens to most, but not all double bonds. So let's say now that we add just 2 hydrogens. When this happens, some double bonds can be turned into single bonds by the hydrogens, but then they might reform. And what you end up with is an unsaturated fat, but some of those fats have trans double bonds. Partial hydrogenation is a process that happens naturally in the digestive tract of some animals, like cows and pigs, which is why trans fats can be found naturally in meat and dairy products. Trans fats are also created through the partial hydrogenation of liquid oils, a process that makes them solid. Partially hydrogenated oils have been largely removed from foods in North America and Europe, because trans fats have been associated with coronary heart disease. Although some foods might have more of one type of fat than another, the truth is that all foods are made up of a blend of fatty acids. When you eat a food like peanut butter, which has about 75% of its calories from fat, the body goes through a set of steps to digest and absorb the fatty acids. First of all, triglycerides are hydrophobic, so they form large globules of fat, like what you see when you pour oil in water. Enzymes called lipases in the saliva, stomach, and secreted by the pancreas can break down triglycerides into free fatty acids and monoglycerides. But working on the surface of a globule is inefficient. So to speed things up, bile salts produced by the liver break the large fat droplet into smaller droplets, which increases the surface area for the lipases to work. Once the triglycerides are broken down into monoglycerides and free fatty acids, these self-assemble into mixed micelles, which have a hydrophobic interior and a hydrophilic or water-loving exterior. The micelles glide through the watery environment of the intestinal lumen and reach the enterocytes in the intestinal wall. When they get to the enterocytes, the micelles release the fatty acids and monoglycerides, which diffuse into the enterocyte. Inside the enterocyte, the fatty acids and monoglycerides reassemble into triglycerides, and these get packed into a larger structure called a chylomicron. The chylomicron has lipids and proteins, so it's a lipoprotein. It has an outer membrane with phospholipids and proteins, and a hydrophobic core that has triglycerides, cholesterol, and fat-soluble vitamins A, D, E, and K. The chylomicron then leaves the enterocyte, but it's too large to get into the endothelial cells, so instead it enters a nearby lymphatic capillary called a lacteal. From there, the chylomicron floats in the lymph and flows into the thoracic duct, and then gets dumped into the blood, essentially bypassing the portal vein. Once in the blood, the chylomicron releases fatty acids and monoglycerides in peripheral tissues like muscle, which use them for energy, as well as adipose tissue which can store them. After delivering the triglycerides, the chylomicron shrinks in size and eventually gets engulfed by the liver. Now, fats play a super important role throughout the body. They have a huge number of health benefits, and those benefits can vary by the type of fat we eat. For example, polyunsaturated fats are precursors for hormone-like molecules called prostaglandins that stimulate endothelial cells that line blood vessels to release nitric oxide. Nitric oxide is a vasodilator, so that decreases resistance to blood flow and in turn lowers blood pressure. Polyunsaturated fatty acids can also help reduce the total and LDL cholesterol, and that's linked to lower rates of cardiovascular diseases like heart attacks and stroke. Long-chain omega-3 fatty acids like DHA and EPA can both help to lower plasma triglyceride levels, which also protects against cardiovascular disease. Finally, DHA is important in the development of eyes and brains of young infants. The impact of saturated fat on cardiovascular health is a little more complex. Generally speaking, it's recommended to keep consumption of saturated fat low, but just like with unsaturated fats, there are different types of saturated fats. Evidence suggests that different types of saturated fatty acids might have different effects on our cardiovascular health. Evidence also suggests that the health impact of reducing or replacing saturated fat in the diet depends on the nutrient that replaces it. For example, replacing saturated fat with polyunsaturated fat has been shown to benefit cardiovascular health, whereas replacing saturated fat with refined carbohydrate has not. Based on this, the National Academies of Medicine recommends consuming 20 to 35% of our daily calories as fat for a 2,000 daily calorie intake. That comes out to between 400 and 700 calories from fat, or about 44 to 78 grams. And it's not just about the amount of fat that we eat. The type also matters. The World Health Organization and the US Dietary Guidelines recommends that less than 10% of daily calories comes from saturated fats, and that trans fat consumption should be kept as low as possible. Alright, as a quick recap, fats are an essential part of our diet and health. Fatty acids are either saturated or unsaturated, and foods with fat always have a mixture of fatty acid types. Healthy diets emphasize mono and polyunsaturated fats over saturated fats, and trans fats are associated with an increased risk of cardiovascular disease. Improvements to our diet and health can be made by focusing more on the type of fat we eat and less on the amount.
The molecular structure of a typical amino acid. So the basic structure of amino acids is that they consist of a carboxyl group, a lone hydrogen atom, an amino group, and a side chain. This side chain is often referred to as the R group. The formation of the side chain is what makes amino acids different from one another. As you can see in this diagram, these four are all connected to a carbon atom, which is referred to as the alpha carbon. Now, not every amino acid follows this exact structure, but most do. On the screen I have three different amino acids: lysine, tryptophan, and leucine. You can see that each has a carboxyl group, an alpha carbon, an amino group, and an R group that is different from one another. There are a total of 23 amino acids that are proteinogenic. Proteinogenic amino acids are precursors to proteins, which means that they are compounds that participate in a chemical reaction to produce another compound. Of these 23 amino acids, 20 of them are called standard amino acids, and the other 3 are non-standard amino acids. These are listed on the screen. In this video, we're going to focus on the standard amino acids, as they are what make up proteins. These amino acids can be classified many different ways. We're going to classify them in a basic nutritional way: essential and non-essential. Essential amino acids cannot be made by the body, so they must come from foods we eat. Non-essential amino acids are amino acids that our bodies can produce even if we don't get them from the food we eat. There are 9 essential and 11 non-essential amino acids. I've listed them on the screen. So how do amino acids form proteins? Well, proteins are built from the 20 standard amino acids. The first thing that happens is that two amino acids come together to form what's called a peptide bond. A peptide bond is when the carboxyl group of one amino acid bonds with the amino group of another amino acid, as you can see here. If you notice, two hydrogen atoms and one oxygen atom have been removed from the peptide bonding process. So the peptide bonding results in the release of a water molecule, H2O. But we're not finished. More amino acids can link in and form what's called a polypeptide chain. Some proteins are single polypeptide chains, and other proteins have polypeptide chains linked together. Keep in mind that not all protein contains all 20 of the standard amino acids. Proteins are often labeled as complete or incomplete protein. A complete protein is a protein source that contains a sufficient quantity of all 9 of the essential amino acids that we discussed earlier. An incomplete protein does not contain a sufficient quantity of all 9 of the essential amino acids. Complete protein foods include animal foods such as red meat, poultry, pork, and fish, eggs and dairy products such as cow's milk, yogurt, and cheese, plant foods such as soy products, black beans, kidney beans, pumpkin seeds, quinoa, pistachios, just to name a few. You can also combine incomplete protein foods to create a complete protein meal, or to get the essential amino acids throughout the day. Amino acids also make up most enzymes. These enzymes are proteins, so they are made by linking amino acids together in a specific and unique order. This chain of amino acids then forms a unique shape that allows the enzyme created to serve a single specific purpose. Enzymes function as catalysts, which means that they speed up the rate at which metabolic processes and reactions occur. Amino acids can also be metabolized for energy. Some hormones like epinephrine, also known as adrenaline, are amino acid derived. Some neurotransmitters like serotonin are derived from amino acids. The amino acid arginine is a precursor of nitric oxide, which helps regulate blood pressure, improves sleep quality, and increases endurance and strength. Glutathione, which is a powerful antioxidant, is also formed from amino acids.
If you have been studying the way the body works, you might think about proteins found inside of you and the different roles they play. There are many different types of proteins, and they are the true workhorses of the body, needed for building structures and maintaining life. Yet all proteins have things in common. For example, break any protein down and what you will find are a bunch of amino acids bonded together. This is because amino acids are the basic building blocks of proteins. There are 20 different amino acids needed by your body. Some of them are made in your body while others must come from your diet. All of the amino acids have the same basic structure. We see a central carbon atom, which is represented by the letter C, bonded to 4 different components. We see a hydrogen atom, which is represented by this letter H. We also have a carboxyl group, which is the COOH seen here, and an amino group, which is this NH2 on the other side. So we have a bunch of C's, some O's, H's, and N's in the basic structure of an amino acid, which means the most common atoms in amino acids are carbon, oxygen, hydrogen, and nitrogen. But you probably also notice the R group. This represents a side chain, and it's the side chain that makes the 20 amino acids different from each other. However, what we're concerned about in this lesson is how the different amino acids come together to form protein. We mentioned that some of the amino acids are made by your body, while others come from your diet. Your digestive system takes in these protein-containing foods and breaks them down into the individual amino acids, which are then taken up by your body cells and reassembled into new proteins. Inside your cells, the individual amino acids can bond together by forming a peptide bond, which is simply a chemical bond that joins amino acids together. More specifically, peptide bonds join the carboxyl group of one amino acid with the amino group of another. For example, this amino acid called glycine can bond with this amino acid called alanine to form a dipeptide. The prefix di means two, so it's easy to see that a dipeptide is defined as a compound consisting of 2 amino acids. Now, amino acids cannot just join together by holding hands. They actually have to give something up in order to live their lives together. In our dipeptide example, we see that glycine gives up an atom of hydrogen and an atom of oxygen, while alanine loses an atom of hydrogen. Together, they release a molecule called H2O, and I bet you recognize that as a water molecule. This loss of water to form a bond is called a dehydration synthesis reaction. This is a big term, but it's easy to remember if you recall that dehydration is the loss of water, and synthesis means to build. So a protein is built through dehydration synthesis. Of course, simply joining two amino acids together is still a long way off from having a protein. More amino acids need to join up until you have a chain of amino acids joined together by peptide bonds, which gives us a polypeptide. Polypeptides are important to the body and can be found in places like the oxygen-carrying hemoglobin of your blood. Now, proteins, well, they are also long chains of amino acids, and there's some debate over the distinction between a small protein and a polypeptide. Yet we do see that some larger proteins can contain many polypeptides folded and coiled together. Let's review. Proteins are the true workhorses of the body, needed for building structures and maintaining life. The basic building blocks of proteins are amino acids. The 20 different amino acids needed by your body all have the same basic structure, which is a central carbon atom attached to a hydrogen atom, a carboxyl group, an amino group, and a variable R group or side chain that gives the amino acid its uniqueness. A peptide bond is a chemical bond that joins amino acids together, where the carboxyl group of one amino acid bonds with the amino group of another. This forms a dipeptide, which is simply a compound consisting of two amino acids. It requires the loss of water to form a bond, which is a process called a dehydration synthesis reaction. More amino acids can join up in a similar fashion until you have a chain of amino acids joined together by peptide bonds, which results in a polypeptide. Proteins are also long chains of amino acids, and some larger proteins can contain many polypeptides folded and coiled together.
Proteins play countless roles throughout the biological world. Some transport nutrients throughout the body. Some help chemical reactions to happen at faster rates. Others build the structures that make up living things. Despite this wide range of functions, all proteins are made out of the same 21 building blocks, called amino acids. Amino acids are made of carbon, oxygen, nitrogen, and hydrogen atoms, and some contain sulfur atoms. Selenocysteine is the only standard amino acid that contains a selenium atom. These atoms form an amino group, a carboxyl group, and a side chain, all attached to a central carbon atom. The side chain determines an amino acid's properties and is the only part that varies from amino acid to amino acid. Hydrophobic amino acids have carbon-rich side chains, which don't interact well with water. Hydrophilic, or polar, amino acids interact well with water. Charged amino acids interact with oppositely charged amino acids or with other molecules. Primary structure. The primary structure of a protein is the linear sequence of amino acids as encoded by DNA. The amino acids in a protein are joined by peptide bonds, which link the amino group of one amino acid to the carboxyl group of another. A water molecule is released each time a peptide bond is formed. This linked series of carbon, nitrogen, and oxygen atoms is the protein backbone. Protein chains often fold into two types of secondary structures, alpha helices and beta sheets. An alpha helix is a right-handed coil stabilized by hydrogen bonds between the amine and carboxyl groups of nearby amino acids. Beta sheets are formed when hydrogen bonds stabilize two or more adjacent strands of amino acids. The tertiary structure of a protein is the three-dimensional shape of the protein chain. This shape is determined by the characteristics of the amino acids making up the chain. Many proteins form globular shapes with hydrophobic side chains sheltered on the inside, away from surrounding water molecules. Membrane-bound proteins have hydrophobic amino acids clustered together on their exteriors, so that hydrophobic side chains can interact with the lipids in the membrane. Charged amino acids allow proteins to interact with molecules that have complementary charges. The functions of many proteins rely upon their three-dimensional shapes. For example, hemoglobin forms a pocket to hold heme, a small molecule with an iron atom in the center that binds to oxygen. Quaternary structure. Two or more polypeptide chains can come together to form one functional molecule with several subunits. The four subunits of hemoglobin cooperate so the complex can more easily pick up oxygen in the lungs and release it in the body. Different visual representations of proteins can give us visual clues about protein structure and function. This space-filling diagram shows all of the atoms that make up this protein. This representation, called a ribbon or cartoon diagram, shows the organization of the protein backbone and highlights alpha helices. This surface representation shows the areas in the protein that are accessible to water molecules. Most proteins are smaller than the wavelength of light. For example, the hemoglobin molecule is about 6.5 nanometers in size. Hemoglobin is found in high concentration in red blood cells. A typical red blood cell contains about 280 million hemoglobin molecules. The three-dimensional shapes of proteins determine their function. The flexible arms of antibodies protect us from disease by recognizing and binding to pathogens and targeting them for destruction by the immune system. The hormone insulin is a small, stable protein that can easily maintain its shape while traveling through the blood to regulate blood glucose level. Alpha amylase is an enzyme that begins digestion of starches in our saliva. The calcium pump is aided by magnesium and powered by ATP to move calcium ions back to the sarcoplasmic reticulum after each muscle contraction. Ferritin is a spherical protein with channels that allow iron atoms to enter and exit depending upon an organism's needs. On the inside, ferritin forms a hollow space with iron atoms attached to the inner wall. Ferritin stores iron in a non-toxic form. Collagen forms a strong triple helix that is used throughout the body for structural support. Collagen molecules can form elongated fibrils, which aggregate to form collagen fibers. This type of collagen is abundant in skin and tendons.
Vitamins are non-caloric micronutrients that are needed in small amounts for normal body functions. The best way to consume vitamins is through a variety of nutrient-dense foods. Supplements can be used to treat certain health concerns, but there's insufficient evidence of any nutritional benefit in healthy people. However, if supplements are consumed, choosing a whole food-based vitamin may be a viable option. There are a total of 13 vitamins, which are divided into two categories: water-soluble, which includes vitamin B's and C, and fat-soluble vitamins, A, D, E, and K. Water-soluble vitamins cannot be stored in the body, except for B6 and B12. and are readily excreted if not used. Because water-soluble vitamins are not stored in the body, consistent daily intake is important to avoid deficiencies. Water-soluble vitamins are necessary for enzymes involved in metabolism. Without water-soluble vitamins, the enzymes will not work. Unlike water-soluble vitamins, fat-soluble vitamins can be stored in the body for extended periods of time and later excreted. This storage usually occurs in the liver and adipose tissue. The ability for the healthy body to store fat-soluble vitamins decreases the risk of deficiencies. However, overconsumption can lead to toxicity, also referred to as hypervitaminosis. When consuming our vitamins through foods, the risk of overconsumption is extremely rare. The risk of toxicity usually occurs through high amounts of supplementation. Minerals are inorganic substances, meaning that they're not derived by living matter, that are needed in trace to small amounts to help regulate bodily functions, such as assisting with muscle contraction and regulating enzyme activity. Nearly all minerals, with the exception of iron, are absorbed in their free form. In most cases, when minerals are bound to other molecules, the mineral absorption is impaired. Also, mineral-mineral interactions can interfere with how well minerals are absorbed. One example is that iron supplementation may decrease the ability of the body to absorb zinc. This is worth noting when considering taking mineral supplements. The degree at which a mineral can be absorbed is known as its bioavailability. High mineral bioavailability can be categorized when greater than 40% of the mineral can be absorbed. The minerals included in this category are chloride, fluoride, iodine, potassium, and sodium. Minerals that have low bioavailability, that less than 10%, include chromium, iron, manganese, and zinc. All other minerals have a bioavailability of 30 to 40%. The minerals that the body needs are called essential minerals. There are 16 of them, which I have listed on the screen. These can be divided into two groups, macrominerals and microminerals. Macrominerals are major elements that include calcium, chloride, magnesium, phosphorus, potassium, sodium, and sulfur. These elements have recommended daily intake of 100 milligrams per day or more for adults. Microminerals are trace minerals that include chromium, copper, fluoride, iodine, iron, manganese, molybdenum, selenium, and zinc. Of these trace elements, the only ones that have established recommended daily intake are iodine, iron, selenium, and zinc. A great approach is to consume a balanced diet that contains a variety of nutrient-dense foods that are not highly processed. That can provide the body with the essential minerals.